Okay, I'm going to be teaching you briefly about blood types. You just finished the blood type donor lab in class, and at this point, you should have discovered who is the universal receiver and who is the universal donor. So we've talked a little bit about this, but blood types we didn't really start understanding until the 1900s, and many people actually died or had serious complications after blood transfusions. Because what was happening is if they were given the inappropriate type of blood, their red blood cells would clump together and it could cause a really bad reaction, even possible death. And so it wasn't until all that started happening that researchers and scientists realized, okay, not all blood types are the same. So now we know that there are 8 main types of blood. So you have type A, then you have type B, you have AB, and then you have O. And each type can also have a positive or a negative form. So that gives us 8 total blood types. And this is actually determined genetically. And this is determined by a marker molecule called an antigen. And you should be familiar with that word because we learned antigens when we studied pathogens. Remember, that was a protein marker on the outside. So red blood cells have a similar structure on their red blood cell on the outside of it that determines and identifies it as a specific blood type. So the way that I'm going to kind of draw this out while I teach this to you is if let's just say that this is type A blood, okay, this is type B blood, this is type AB, and then this is type O. So type A has a special marker on the outside of it that identifies it as type A. Let's say that this is what type A's blood marker looks like. Type B has a different kind of marker. Let's say that this is the type B marker. Type AB has both. And then type O has none. Okay? So these marker molecules on your red blood cells determine your blood type and the type of blood you could safely donate and safely receive. So your plasma, your blood plasma, as we learned that blood is a mixture that has 4 components: red blood cells, white blood cells, plasma, platelets. Okay, so those are the types, the different components that make up blood. And so blood is a mixture of these 4 things. Your plasma is like the water part of your blood, and it is what all of the different nutrients will flow through. So the red blood cells with these markers, if there's a foreign red blood cell in your body will kind of have a reaction to it, and it will start clumping them together if the wrong blood cell is in your body. So here's another picture, like I kind of drew, something similar. This would be, for example, group A. That's what their antigen looks like. There's B. AB, notice, has both, and O has none. And then you've got another diagram here that shows the different antibodies and antigens. I just don't worry about this anti-A and anti-B. Just kind of look at my drawing here. That will seem to make sense. So you also have what's called the Rh factor. This is another protein on your red blood cell. If you have it, then you're considered Rh positive. If you don't, you're Rh negative. So Rh positive blood types also, there's an issue there with being able to donate and receive on the Rh factor. So for example, if you have it, then you're considered positive. So this would be an A positive blood type. This would be a B positive blood type. This would be an AB positive blood type. And then this would be an O positive blood type. Whereas, for example, if you were type B blood but did not have the Rh factor, you just wouldn't have it. So this would be considered B negative, whereas this is B positive. Okay? So the biggest thing to determine universal donor and universal receiver is you don't want to change the red blood cell at all. So you can't make it look any different than what it originally looks like. So it's an inherited trait, and there are 4 basic, you'll remember, phenotypes. This is what it physically looks like your marker molecules. You have A, B, AB, and O. Each has a positive and negative. Type AB blood is actually codominant, which means that it's both expressed in the phenotype. Then you can have what's called big A, big A. You can have big A with a recessive O. You can have big B, big B. You can have big B with a recessive O, or you can just have the recessive type of OO. So this chart is really helpful because this shows you genotype and then phenotype. Okay, so if you have big A, then what type blood do you have? You have type A. If you have big A, little o, you still have type A because A is dominant to O, and then you can see the rest there. So we're going to be doing some problems here to determine what the blood type would be, but we're actually going to ignore the RH factor. That makes it a little bit more complicated. So if we're trying to determine who can safely receive from other blood types, then I want you to picture this. Okay, I probably need to let me just go down to the bottom here. We don't want to change the red blood cell, right? So if this is my A, okay, and I am positive, I have the particular RH factor, and then this is type B, and I look like this, okay? Can B receive types A's blood? If A is given to B, then it would change B to look like this because it would give him those antigens, and that would not be a safe combination. And so in your lab today, you kind of went through that safe and unsafe, so you should have a good idea of what can be given and what cannot be given. And finally, I am going to do some examples of some Punnett square problems of how you would set this up. And then you have a homework assignment that also explains this. So we can determine genetically what blood type you would receive. And so say that you have one parent who is type O blood. Well, type O is the recessive blood type. So the genotype has to be little o. And then you have another parent, say they are type A, but a carrier of O. So this would be their genotype. So setting up your Punnett square, and then we've done all of these before. So we're going to go through. And what's our results here? In this situation, you can have 50% have AO, which would be A type blood. And then you have 50% that are going to be OO, which would be type O blood. And we would have to do a separate Punnett square for the RH factor. Okay, so the RH factor is a separate inherited trait. So for example, if you have one parent that that has the RH factor, okay, and then you have another parent who does not. So the negative RH factor is actually recessive. And so you would have plus negative, plus negative, plus negative, plus negative. So this combination would be 100% of RH positive blood types. So those are just some examples. So you'll need to use this this sort of key to help you do the problems on assignment 6.9. You'll open that up in Canvas, and there's some questions about doing some Punnett squares. Shut this off. I think it's done. Hold on. I got to press my screencast and pause it. Do y'all want to come say hi? Say hi.
Hi. Okay. Okay, so actually say hi.
All right, quickly, I'm going to stop it. Do what you want to do.
Hello.